Okay. So this morning we talked a lot about, well, the basics of how we would analyze the data, and I mentioned several times that one prerequisite for this, or at least maybe not prerequisite, but it certainly helps, is if you have some prior knowledge about the set of peptides and proteins that you're interested in extracting from the DIA data. And so the purpose of this talk is to say something about how we generate this prior knowledge, what we refer to as spectral libraries, or sometimes as peptide query parameters. And so, okay, here we go. So why do we care about prior knowledge in DIA? I think we talked a bit about this in the morning. Second part will be just to kind of walk through the procedure for generating these spectral libraries and peptide query parameters and try to point out a few issues that might come up. And then finally make a few comments about publicly available resources and whether this is it's a good idea to use these or not. So first, why do we care about this? Well, the two main uses that we make of these is one, to direct the software which signals to extract from the data, right? So which fragment ions from which peptides do we want to try and extract from the data if we're going to try and do quantitative analysis of inferred proteins. And the second thing we can do with the prior knowledge is to use this as a reference to score the measured data against, right? So we can gain some kind of power in the analysis by having observed this before and making some comparisons in the DIA data to what we already think we know what these peptides look like to the mass spectrometer. And this is primarily in the form of, well, firstly, which peptides are most observable, but then what are the relative fragment ion intensities in the spectra for these peptides, and what is the chromatographic behavior of these peptides, and this usually has to be done in some kind of normalized space. So I'll describe a little bit about this. And so just to remind you, I already showed this slide earlier, what's the problem? I mean, we can't search these complex MS2 spectra from DIA analysis in the kind of classical way. So we need another approach, and the approach I said we're taking is to look for particular peptides. We need to go in and dig out the signals for the peptides that we're interested in, but this implies that we know something about what this peptide looks like already. So this is the spectra again in this complex background. The fragment ions for this peptide are kind of barely visible there. So we need, if we want to dig these out, we need to know where they should be and what their characteristics are before we try and do that. So that's what's depicted here. So if we input some prior knowledge, here's a spectrum for this particular peptide. This is how the MS2 spectrum looks like. And if we have some confidence that this is how this peptide looks to the mass spectrometer, then we can use this pattern to kind of dig into this complex DIA data and extract selectively the signal that we're interested in. So we go from this reference library through the DIA data and hopefully end up with something that looks like a selective signal. And so when we started doing this a few years back, people used to always ask, well, we would go to meetings and say, if you do this kind of SWATH analysis and you know something about the prior knowledge, and you have some prior knowledge about the peptides in the form of a spectral library, well, this will get you to some high quality proteomics data. And then everyone would say something like, well, where do you get the spectral libraries? There was a lot of confusion about this. And so this led us to try and write this down in a sort of structured way, and we published this protocol paper a couple of years back, which basically says how we do this. And the points, the sort of goals here was to sort of document, a protocol that other people could potentially use if they want to, but also to document how any libraries that we put out into the world are being made in some detail. So if you're interested in more deeply in this topic, I would suggest you take a look at this particular protocol paper. And so I also showed this slide earlier, but just to remind you that this is the goal now. So this is the outcome that we want at the end of this spectral library generation stage, or this peptide query parameter generation stage. So this is the proteins, the peptides that we're interested in, and how they look to the mass spectrometer in terms of their precursors and fragments, relative ion intensities, and their retention time in some kind of normalized space. So this is also what we will do in the tutorial in the afternoon. In the first tutorial will be the spectral library generation tutorial, and the goal that we are going to try and get to by the end of the day is a table that looks like this. Okay, so let's take a look at what this procedure actually looks like. So So I think most people, I guess, in the audience are familiar with DDA analysis and how to do DDA database searches. So that's actually the largest chunk of this procedure, but I just want to make some comments about the specific steps with respect to DDA analysis. So we're going to start with some kind of DDA data acquisition. We're going to do data conversion and peak picking. Usually we typically use these open XML-based formats, but we need to do some centroiding and peak picking for submission to a database search. Then we do the database search. And very important is the false discovery rate control of this database search. And then, so this is the sort of standard DDA pipeline, I would say. And then from here down is a little bit more specific. So what we want to do with the DDA search results is combine these into a spectral library. And this primarily means reducing the redundancy for MS2 spectra. So if we have a given peptide and we have multiple MS2 spectra acquired for that peptide and they're all high scoring, then we only need one representation of that peptide in the library. So we need to reduce that redundancy somehow. And this is typically done in this kind of spectral library step. And then we want to filter this spectral library because we probably don't want to extract all fragmentines for every peptide. So we need to make some decisions about what fragmentines we want to monitor from which precursors. And so this is essentially a filtering step. And then at the end, we have what we usually call peptide query parameters, just to distinguish them from this spectral library, which has the full MS2 spectra here. Here we have basically this table, which is a set of fragmentines that we want to monitor. So this is the easy kind of looking version. This is the slightly significantly more complicated looking version of the workflow, and this is what you kind of find in this protocol paper if you're interested in that. So I don't think we need to really go through this in a lot of detail. This is basically what you're going to do in the tutorials. We use ProteoWizard for file conversion and peak picking. Actually, for peak picking, we use this QTOF peak picker tool. And then we do some search engines. We tend to use Cometa and Xtandem. We tend to use the open source ones here. And we do some post-processing of this and using the Peptide Profit and iProfit tools to combine these. If we have searches from different databases, different search engines, sorry, we combine them using this iProfit tool. Then we have another tool called Mayu, which does all the FDR estimation at different levels. And we select a filter here, and we only let the high quality stuff in to this next step, this library, spectral library generation stage. This is done using a piece of software called Spectrast from the TPP. And here we're reducing the redundancy of the MS2 spectra, but this is also the stage where we're doing this retention time normalization. So I'll say what this means in a few slides, but we basically need to have some standardized chromatographic space where we store the retention times of peptides. And then the final part is basically this filtering. We want to choose a set of fragmentines from these MS2 spectra to monitor. And then there's a bunch of different formats, either in table formats, which can then be imported into Skyline or Spectronaut or other softwares. And for open SWATH, we're using this TRAML format, which is an XML-based format, but actually more recently, we now have something called PQP, which is a SQLite-based format. But yeah, this is the goal, right? So the goal at the end of this workflow is going to be this set of query parameters. And just to mention, this is probably rather a complicated looking workflow, and this is the way we do things in our labs and also the way we're going to do it in the tutorial. But you'll also see from the sessions with Brendan on the Skyline tutorial, there are other alternatives here. So you can also take whatever your kind of favorite search engine is here and plug it into Skyline, and Skyline has a tool called BiblioSpec packaged with it, which does the spectral library part, and it also has functions to do all this filtering. So there are kind of alternatives here, and we'll probably try both of them. So the next question is then, what kind of samples do you want to actually put into your DDA analysis for the spectral library generation? And there's some choices to be made here. So of course, the simplest thing is just to take the samples of interest, so the same samples that you're going to acquire DIA data on, and analyze those by DDA. The issue is that our experience has been this sort of direct comparison of single-shot analysis of DDA versus DIA. You tend to have better sensitivity and coverage in the DIA data, and therefore, so the point is that a just a simple single-shot replicate analysis of the given sample of interest is probably not going to generate a sufficiently large spectral library to completely exploit all the information that's in the DIA file. So sometimes what people will then do is take these samples of interest and do some fractionation. So they'll do various types of peptide fractionation, like high pH reverse phase seems to be people's favorite one these days, and they'll get a deep kind of proteome coverage DDA library of the sample that they're interested in. But there are also other alternatives. So in our lab, we used to synthesize a lot of peptides in this kind of dirty synthesis mode this solid phase spot synthesis mode where you would synthesize a lot of peptides in 96-well format for like $10 or $20 per peptide. This can also be useful. Other labs have gone for like full-length recombinant proteins or synthetic proteins. This also has its advantages, and we'll talk about that later on. Or you can go out into the public repositories and see what's available for the proteins that you're interested in various types of repositories. And these repositories range from like just large-scale ones that are for a given what I call organism scale libraries, or there's much more focused repositories like the CPTAC or Panorama, where there's very refined assays for particular peptides. But there's certainly stuff out in the wild which you can potentially use. And these, or you can use some combination of these. And the idea is that you will now generate the Spectral library and go through to get this table of peptide query parameters, which I mentioned. This is another figure stolen from this SWATH MS tutorial, which I mentioned from Tina Ludwig, that should come out in the next few weeks. So so basically we're at the start here. We're deciding what kind of samples we want, and now we need to do the DDA acquisition. So there's not much to say here. I would make a few comments that is there anything different that you would do with respect to a standard DDA analysis? We've noticed that there might be some benefit, for example, to increasing accumulation times to get higher quality MS2 spectra. So for example, on the platform we use the most, sometimes we boost the MS2 scan time from like 50 milliseconds, which is the scan time you might want to use for maximizing IDs, but sometimes we push this up a little bit higher. And the benefit of this is that you get a bit better signal to noise in the MS spectra, and the representation of the relative fragment ion abundances. So like if this is, this Y4 ion is kind of 20% higher than this Y5 ion, and this ranking and the representation of these relative intensities is essentially truer the higher quality you have for your MS2 spectra. And this can be this fidelity of matching can be improved by going with a little bit longer MS2 scan time. This is particularly in TOF. It's probably less important in Orbitrap data. Sometimes something else we do, which also people have done for a while in QTOF type analysis, would be to try to sample more than once a given precursor with MS2 spectra. So you can do tricks like kind of setting the dynamic exclusion to 1/3 or 1/2 of what you expect the chromatographic peak width to be, such that you sample maybe 2 or 3 times across the elution of the peak, and this allows you to have more representations, more instances of MS2 spectra for a given peptide precursor, which might kind of go in the same direction, that you will end up with a more true representation of the relative fragment ion intensities in your final spectral library entry. The other thing we occasionally do is we apply, because in DIA mode, we can't very finely control the collision energy because we're co-isolating peptides which have different charge states. We can't specify, we can't customize the collision energy for all of those charge states at the same time. So what we tend to do is use the collision energy for 2+ precursor ions. This has the downside that probably higher charge state precursors are being over-fragmented, which is not a great thing, but something you can do to kind of compensate for this in some way is when you're collecting the DDA data, you can apply those same set of collision energy rules to the DDA analysis. And then at least the matching between the DDA relative fragment ion intensities and the DIA will be coherent. There'll be sort of higher fidelity matching. This might come at the cost of reduced ID rate, and in fact all of these measures might come at the cost of reduced ID rate in the DDA data, but at some point might increase the spectral quality that allows you to have a higher quality spectral library, and it might benefit you in the end. And this is especially true, I think doing things like this is especially true if you're taking the option to go for like fractionated samples of interest. Then this library is probably going to be a lot deeper than what you can actually extract from the DIA analysis. So maybe it's worth trying to improve the quality of the spectral library at the cost of losing some IDs, because you might not need all of those IDs anyway to exploit the DIA data. So we normally do some conversion then into these kind of open formats for searching, but what I'm more interested in pointing out here is that we do some peak picking and centroiding at this stage because the search engines require a peak list to search against. And I would just show one slide to point out that it's worth knowing what your peak picking tool is doing, because sometimes they're doing more than you expect. So this is an example of a single MSMS spectrum which has been converted and peak picked using 3 different tools. So this is the ABSciX tool, ABSciX converter. This is MS Convert, which is using the sort of vendor option so it's the ABSciX API, which is doing the peak picking. And I should say that this is now performing better than it is in this picture, but I just show it for the purpose of illustration. And then this is a tool called QTOF Peak Picker, which was developed in our lab. And so this is the same raw MSMS spectrum, right, but processed in 3 different ways using 3 different peak pickers. And what you see is you get a very different, potentially a very different representation of the relative fragment ion intensities. And sometimes you can even have things happening like there's charge state deconvolution going on where multiply charged ions might be deconvoluted back to single charge state and would be in completely the wrong place. And so this is the reason why we're using this QTOF peak picker a lot, because it does relatively little processing to the spectrum. And what you end up getting is a good match between the relative fragment ion intensities that we see in the DDA data and the relative fragment ion intensities when we actually go to the DIA data and extract these out, right? So here you can see this Y10 ion is the most intense in this one and also this one, but not in these guys. So just be aware of what any kind of peak picker you're using might be actually doing. So then we need to do a search, and so I think I guess everybody knows this and probably has their own kind of favorite search engine. So we're tending to use these as well as a few other of the kind of open source ones, and we process these through Peptide Profit and iProfit. And this iProfit tool is the one that basically allows you to combine search results from several different search engines. And this is potentially useful because it's just boosting a little bit the sensitivity. So if you look at, as a function of sort of fixed FDR, the number of correct PSMs, when you take these 5 search engines and you combine them individually or you combine them together, you get basically better discrimination. So by kind of leveraging all these together, you somehow get a better discriminant score that allows you to have a little bit better sensitivity at fixed FDR. This isn't necessary. This is just a kind of an optimization that we tend to use. So FDR control is also very important, right? So it's at the spectral library generation stage, it's also important to think about this, right? It's not only at the DIA. We'll have lectures tomorrow, I think, talking about FDR control when you're extracting DIA data, but you also already need to think about this at library generation stage, because it's possible that any errors that you make at this stage will be sort of propagated forward into the DIA analysis. And so Rudy, I think it's interesting to think about what might happen here. So let's imagine that you have a particular spectrum, and for whatever reason in the DDA analysis, the wrong sequence was assigned. It was not very informative, but somehow it sort of passed your threshold. So the wrong peptide sequence is assigned here. And the question is, what happens when you go to the DIA data? Well, if this fragment ion spectrum is arising from like a single peptide, a single species, then you will just basically propagate this error into the DIA analysis, because you will get a very nice looking signal group and all the fragment ions will coalute, and the relative fragment ion intensities here will match very beautifully to whatever is in the spectral library, but it's basically just a propagation of this error into the DIA data analysis. It's possible that some types of errors that occur in the DDA analysis would be resolved. So for example, here's a sort of a chimeric spectrum where the fragment ions are arising from 2 different precursors, and that's the reason why the search engine now has misassigned this sequence. So here we have a mistake again in the DDA analysis, but for a different reason than over here. And now if we go to the DIA data, well, you hope that actually this might be resolved because these precursors might not coalute, right? So then the open SWATH, for example, will be trying to reconcile that all these fragments come from a single peptide precursor, but actually they're separated in retention time, and then it would get a bad score, and this error would essentially be resolved. But the problem is we don't actually really know what's the prevalence of these different types of errors. So it seems like the position we should take is to make sure that we have relatively high quality DDA data for spectral library building. And so Rudy showed a picture a bit like this one earlier on, just making the point that there can be differences between the PSM level, the peptide level, and the protein level FDR. So if we plot as a function of this iProfit score, which is essentially a search engine score, you can think about it where the best hits are over here. And if we plot the FDR at different levels, either PSM, peptide, or protein level as a function of this score, what you see is that at a fixed score, we can have, you know, at 5% protein FDR, we can have much lower false discovery rates at the PSM or peptide level, or rather, we should probably think about it the other way around, that at 1% PSM level FDR, we can be up above kind of 10% protein level FDR. And this is because of this phenomenon that Rudy mentioned, that these PSMs, so these green PSMs are accumulating, and in this depiction, the hexagons are the proteins, right? So the correct ones all build up on some relatively small number of proteins, but the false ones are basically distributed over the proteome. So at the protein level, you can accumulate these in a very fast way, and it's just what pointed out here is that we can have different false discovery rates at different levels. So we tend to control these at 1% protein FDR, and this gives us like, well, in this data set, it would give us a peptide FDR of 0.2% and a PSM level FDR of 0.08%. So this is fairly conservative, and the goal here being not to propagate too many errors, not to have too many errors in the spectral library so that they are not propagated into the DIA analysis. Then we need to do some spectral library generation. So we have our DDA search results, and there's two things happening at this stage. One is retention time transformation into a normalized space. And the second thing is that we need to do this reduction of redundancy in the spectral library, right? So let's look at retention time first. So actually, there was a question already after the first lecture. How can we deal with, you know, I said we're using the chromatographic behavior of the peptides as prior knowledge to do later DIA analysis, but what if we're using a different chromatography system or a different column or whatsoever? And so what we're using here is the idea of a retention time index. So if this is the analyte you're interested in, what we do is we spike in a bunch of standards into the sample. So in this case, it's 11. The set we use is these IRT peptides from Biognosys. This is 11 peptides, which we spike into all the samples. And now what you can say is instead of having the absolute retention time for the peptide that you're interested in stored in minutes, you can instead say, oh, I'm going to store an index, basically. I'm going to call standard A as 0. I'm going to call standard B as 100. And I'm going to say the analyte that I'm interested in is 60% of the way between standard A and standard B. And so now we have an index which is no longer stored in minutes, but rather in some unitless space. And this is all relative to these spiked in standards. And so if we now spike in all these standards to all the runs we do, including both for the DDA analysis and later for the DIA analysis, then we can use the chromatographic information that we got from one run to derive confidence in the peptide detection in another run. So here I'm showing this set of peptides that we use from Biognosys, but by now there are many other similar sets of peptides available commercially, and this is kind of the chromatographic behavior of those peptides. And we have basically a fixed number in this, an index, which we assign to each of these peptides. And confusingly, it doesn't go from 0 to 100. It goes from -24 to 100. So that's because they realized this early peptide is a little bit more variable than the others. So they decided to set the second peptide as 0 and the final peptide as 100. But actually, the scale of these numbers doesn't really matter at all. Except that it's a good idea to probably keep using this scale, because then that's what other people in the community are using. So how does this actually work in practice? Well, what we do is we're going to do some DDA analysis of the samples that we're interested in, and we're going to spike all of these 11 peptides into all the samples that we acquire in DDA mode. And what we do is we make a regression, so we plot the measured retention times for these 11 peptides in a given run, a given single DDA run, and we make a regression of that with the IRT values, which are coming from this table here, and these are just fixed values in this unitless scale. And then for all the other peptides that we identify in this DDA run, so these are the peptides now that we're interested in, peptides A, B, and C, we just project through this regression to get values in this IRT space. So now for this peptides A, B, and C, we have a measurement of their chromatographic behavior, but it's in this normalized space where we don't need to worry about absolute retention time. It's just essentially in an index which is defined by this set of standards. And in our spectral libraries, we store the chromatographic retention time values for these peptides in this unitless space, which we call IRT. And then when we come to actually go, oh yeah, and this is this final column of this library that I said that we're trying to get to by the end of this exercise. And you can see that some of these actually have negative values. That's because of this interesting choice that was made to have this first peptide as negative, but it doesn't matter. It's just it's a unitless number. And this is what we carry around in these spectral libraries, these retention times in this unitless space. So now when we actually go and do some measurements, we do some DIA measurements, we can do essentially this exercise in the reverse order. So we have, again, spiked these 11 IRT peptides into the DIA run. And notice that this can be a different gradient, right? So here we've got like a 70-minute gradient. Here we've got a 35-minute gradient. And we measure the retention times of these IRT peptides. Basically, we construct a regression based on the measured values of the IRT peptides in this particular run versus the fixed scale for the IRT peptides that we've defined. And what we can then do is then take our target peptides of interest, these A, B, and C peptides, and we can project it back through this regression to get predictions about where we think our peptides of interest are going to elute in this particular 35-minute gradient as opposed to the 70-minute gradient we use for the library generation. And so this is the procedure that's happening all the time, and you're going to do the first half of this in the first tutorial today, and you'll see more of it later in the week. And so this allows us to do two things. Actually, we can cut down on the computation time because we only need to extract ion chromatograms in the retention time space where we think that these peptides are actually going to be with some tolerance. But we can also calculate a score, which is the delta from where this peptide appears in this retention time space of this one with respect to where we predicted it to be, right? So the closer it is to the predicted value, the more likely it is to be the peptide that we're actually interested in. So the second thing we need to do during this spectral library stage is is removal of redundant spectra. And so I guess the idea here is that for this particular peptide sequence, we do 3 DDA runs, and it's a bit higher or lower in intensity in these different runs. So in this first one, we got 2 MS-MS spectra, which were triggered here and here, and we identified this peptide from these 2 PSMs. Here, it was a bit lower down in intensity, so it never triggered an MS2 spectrum, so we have nothing here. But in this third one, for whatever reason, we just got a single trigger, and we now have 3 MS-MS spectra, which are presumably are all reasonable quality, but we don't need 3. We only need 1. We only need 1 representation of this peptide. So we need to do some kind of redundancy reduction. And there's basically 2 choices here. So you can either just say, the kind of simplest thing to do is I'm going to take the best replicate spectrum, so the highest scoring in terms of the search engine score. I'm going to take that and I'm going to throw away all the other ones. Or you can make a consensus, right? So you can do some kind of averaging and average over these different spectra and get a consensus spectrum. And this is actually one, so we have a little bit of evidence from prior work that this consensus idea is giving you a spectrum which in the end agrees better with the DIA data. So this is some measure of, like, for example, a dot product score where we compare to the DIA data and we get sort of higher fidelity matches when we go from either taking the best replicate spectrum up to the top 5 or combining the top 10, and then it eventually saturates. So now this is something me and Brendan always argue about, because in Skyline they use this best replicate strategy, or in BiblioSpec rather, and we use this consensus spectrum approach, and we haven't really figured out who's right yet, but this is one place where you get a slightly different behavior. Okay, so now we have these spectral libraries where we have one MS2 spectrum for every peptide precursor that we're interested in, and then we just need to do a little bit of filtering to say we typically take we've found that around 6 fragment times per precursor gives us pretty good sensitivity and selectivity trade-off. There's no reason really to restrict the fragmentines. We go in this mass region here, so we don't usually go down, although we measure from like 50 to 2000. Normally, we don't take typically fragmentines below 350 or 400 because they tend to be in a very noisy part of the MS2 space, and they're not very informative anyway. They're not very selective fragment ions. We take B and Y ions usually, unless there's some heavy labeling reason why we can't, for example, use the B ions. We generally would exclude fragment ions that are situated in the precursor isolation window. So you can imagine that in the precursor isolation window, let's imagine it's like 500 to 525. We tend to take fragment ions that fall outside of that range because this precursor isolation window tends to have a lot of unfragmented precursors. So this tends to be also a very noisy part of the spectrum. So we would usually kick those out too. And then we convert these to these formats, which you're going to see later, either PQP, which is the slightly newer way of doing things, or TRAML, which is the older way of doing things. So this is SQLite and this is XML, but that doesn't really matter. And we also need to add some decoys. And so the generation of the decoys and why they're important is going to be covered tomorrow, I think. And so we end up with this, essentially what's a table, and we also have a table format, but basically we can import this table into any, and now there should be actually a lot more different kinds of software here, and we can start to do some analysis of the DIA data. So just to kind of summarize this whirlwind tour of spectral library generation, this is the sort of overall workflow, which looks very much like a standard DDA search until you get to down here where you have to do a few extra steps in order to make this into a spectral library, and then finally a set of peptide query parameters. So we need to make some choices about what kind of samples you want to use for this. Representative samples are probably a good start and a safe thing to do before you get going, but there are other options. It's nice to have good representation of the relative fragmentine intensities in the spectral library from the DDA data, and it might be in some cases even worth kind of hamstringing the DDA analysis in order to improve this such that the spectral library information is similar in its characteristics to the DIA data. So watch out for these peak pickers, which might be doing weird things that you don't know about. Better quality spectra will agree with the DIA data better. And well, again, we do this consensus library making, but best replicate also works very well. Definitely have some way to control the false discovery rate of the DDA data. Otherwise, you're going to be propagating errors into the DIA data. And it's good to transform this retention times into this normalized retention time space using standard peptides like IRT. And in fact, it's also possible to do this without using standard peptides if you've there are kind of endogenous peptides which are in most samples that allow us to do these kinds of transformations as well if you don't want to spike these in. Okay. So yeah, I would point you to this protocol paper if you're interested in looking at this in more detail, but this is now what we're going to do in the tutorial. Okay, so the last topic is, what about these kind of large scale or publicly available spectral library resources? Is it a good idea to do this? And so let's talk about this a little bit. So at some point, we realized that we had this kind of SWATH project 1. We wanted to do data analysis number 1, so we needed kind of spectral library number 1. So we'd do a bunch of DDA analysis on the samples of interest. And then we would have numbers 2 and 3. And then eventually we said, well, maybe we should try to make a collection of these spectral libraries and just consolidate these, and hopefully we might be able to eventually leave this step out of this particular pipeline if we just accumulate enough information. And so this is an example of that. So this is at the time when we did this we decided we would make a spectral library for all of the human DDA samples that we happened to have in the lab at the time. And this is now a few years ago, so it was just it was roughly 300 samples that we had analyzed on our TripleTOF platform, and this came from a lot of different places, different cell lines, blood, some tissues here and there. Some of these had like fractionation, some of them not. Some of them had affinity purification and all sorts of stuff, and we just packed them in all into a single search. And we went through this kind of protocol as I described it, and the results were something like this. So we came up with around 10,000 proteins. about 140,000 peptides, and about 200,000 precursors. So the number of peptides and precursors might seem kind of low with respect to the number of proteins, but remember the way I said we're doing the FDR control, we're very strict at the peptide and precursor level. So essentially what happens is that each one of these peptides and precursors has to independently by itself be able to achieve a 1% protein FDR. So this is a strict interpretation at the kind of peptide and precursor level. And so if you look at everything that's in the canonical Swiss prot, which is the big pink circle, which is around 20,000 proteins, our coverage was about 10,000 or so. And then the green circle was the portion of Swiss prot that had protein level evidence from other places. And this was published now, I guess 4 years ago, but we still use this library sometimes because it's convenient. And the place where we distribute this, or actually this is hosted by our colleagues in ISB in Seattle. There's this SWATH Atlas portal, and we have this human protein library there, but we also have similar protein libraries for various different species and different kinds of samples. There's a HLA peptides one. We have a very comprehensive tuberculosis library because we work on tuberculosis quite a bit, and that consists of like fractionated lysates, but also a whole bunch of synthetic peptides. So we get very kind of high coverage in this particular library. And there's other libraries with other degrees of coverage. And they are kind of downloadable in these formats, and you can try them out if you want. The question is whether you actually want to do that. Other than that, there's also this SRM Atlas, which contains all basically synthetic peptides, both for yeast and human. And this is a project which is mainly directed at SRM. And there's now this much larger resource from Bernhard Kuster, where he's going to do, well, basically, I don't know if you add up all those numbers, it's a large number. I think they have the tryptic peptides out already, and at ASMS they were talking about various other peptides. And so these are also very large resources of spectra that are potentially useful. Some of the problems with these synthetic peptide resources is that you have to decide in advance which peptides you want to synthesize for which proteins. And this can actually be a problem because there are tools to predict which are going to be the best ones, and they actually turns out not to work very well. And so this is what was used for this SRM Atlas. In the Kuster case, I think he's just doing it a little bit more brute force and synthesizing a lot more peptides, and I think that'll eventually will probably overcome that problem. But this is kind of the two, I would say, large-scale synthetic peptide resources that are out there. Oh yeah, and if those of you who are at ASMS this year would have heard probably several talks about using these types of synthetic peptide library resources as training sets for doing kind of deep learning and prediction of what fragment ion spectra should look like for peptides. And it seems like, or at least my impression was that people have tried this in the past, but this seems to be now the first time where this is actually starting to work properly because, and I think it's because of the very large scale of the training data set. So it will be interesting to see how this continues to develop. I also mentioned that there's much more kind of curated assay libraries or spectral library, well, assay library resources in this case. So one is Panorama Web, which is associated with Skyline, and this is like user curated, deposited assays, and these can be very well refined, and also from the McCoss Lab, just as an example of one resource where they're using full-length proteins. So what they do is in vitro transcription translation of full-length proteins, and then they analyze these proteins. And this has the benefit that you get around having to do any predictions about what the best peptides might be, because you have basically all the peptides, and you should just see what the best peptides are. Okay. The question is whether you can actually use these external spectral libraries. People always ask, well, can I use a library made in this other lab to analyze my data? The answer is maybe. It's not very satisfying, but it depends. So this is a figure from this proteome tools from Bernhard Kuster, and here they do some comparison from QTOF data from our lab and this proteome tools beam type CID on an Orbitrap with a particular collision energy. And they do this analysis at a very wide range of collision energies. And what they find is if they pick one collision energy, it actually looks pretty much like the setting that was on this QTOF. So we have a very high similarity between now this QTOF and this Orbitrap HCD spectrum. And this is now a summarization of that whole data. And basically what they say is if things are kind of comparable in terms of the collision and so on, then you can get actually very high correlation between this TOF data and this Orbitrap data. And there's some peptides that actually don't correlate very well, and they claim that these are probably false positives in this data because they think there probably aren't many false positives in the synthetic peptide data, which actually makes sense. So there is, if you take kind of care, then you can make some equivalence between these different types of data sets, it seems like. What about retention time? Well, this is now, these are now some retention time error plots for large sets of peptides detected in 3 data sets. So Rudy, I think, mentioned earlier, I don't know if he did, but at one point we did a study where we sent out one set of samples to 11 different labs, and we asked them to analyze these in SWATH mode. And these are the retention time errors, right? So I said from IRT we make a prediction where the peptide should be in retention time space, and that should be at 0. And then when you actually detect it in the DIA analysis, well, maybe it's actually over here somewhere. So the delta between these is actually something we score in the software. And you can see that here's the data from us using a 30 centimeter packed emitter with this particular stationary phase. And the library we were using for the analysis of all 3 data sets was one library that we generated in Zurich. And so we have kind of like the best, the lowest error in terms of the difference between the actual chromatographic retention time versus the predicted chromatographic retention time. But you can see in these other 2 labs where they were using also a 30 centimeter column, but a different stationary phase, and it was on this kind of exigent chip LC setup. So it's actually 2 columns, 15 centimeter columns in serial. So it's kind of a different setup, but they still get a pretty similar distribution of the error prediction versus the actual. And then you can see in this particular site, well, they had some troubles towards the end of the week, and for some reason their chromatographic stability was worse in later on in the data, but still the predictions are kind of reasonably not that far away from what we had in Zurich. So what I want to the point I'm trying to get across here is that this is not a yes or no question. Can I transfer libraries around the place? I think this is a kind of shades of gray answer, right? So you can imagine that if you had the same column and you did serial injections, you built your library, you know, in the injection before, and then you did the DIA analysis afterwards, then in this retention time error distribution, you would have a very small error. Well, if you went to now the same stationary phase, but a different column, then you would get a bigger error. And then now you have a different stationary phase, or maybe a different lab, and you're going to have another bigger error. So that means that this information might still be useful to a degree. It's just not as useful as this information, right? So these errors are part of what's scored in the software, and so bad information might be better than no information. But directly comparable libraries from the same instrument, same chromatographic system will give you the best fidelity here. And you can say a similar thing about the spectral similarity, right? So if we have like the same instrument and the same collision energy settings, then in this score where we compare the relative fragment ion intensities, we should have very high fidelity here. And maybe if, as in this case that Kuster showed, if we go from QTOF to Orbi HCD, well then this distribution shifts a bit. And if we look at beam type CID versus resonant CID, like on an ion trap, well then this gets considerably worse. But again, it may not be worse than no information. And so I think there's also an important question. Should I go for the biggest available library, right? So should I just take all the publicly available libraries for my organism and try to extract every peptide that's ever been observed in a mass spectrometer? And I think that the answer is definitely not, because of what Olga just talked about, so multiple testing corrections. So let's imagine we have a DIA data set and we have options of using different kinds of libraries. And let's say this is the set of DIA detectable peptides, which of course we don't know what this is in advance, but let's just imagine that this is the set of peptides which you ever have a chance of detecting in this DIA data. So the ideal library to extract would be exactly this set of peptides, right? So the ideal library would be the very same set of peptides which are detectable. Then we'd be able to detect all peptides in the library. Everything will be great. But unfortunately, we don't know this in advance, right? So the trick is actually trying to figure out what this is. So you can do a single shot analysis of the same sample that you have, and you'll get a library which looks something like this. There'll be some overlap between this kind of ideal library, but you might get a few extra things that were not detectable, that are not detectable in the DIA data for some reason, but there's a whole chunk here that are going to be missing, and so you're not, this is what I was trying to say, that you're not fully exploiting what's available in the DIA data. So then you say, okay, I'm going to fractionate my library into 24 fractions, I'm going to shoot those by DDA and make a bigger library. So now here's this fractionated DDA library, so that should sort of mostly subsume the single shot one, and we increase the chunk of this sort of ideal library that we're getting to, but there still might be something there for some reason that you don't get, that you have in this fractionated data that you don't necessarily get in the DIA data. You could go now to the organism scale repositories, and now you cover more of this maybe. But the point I want to make is that we can never identify anything more than what's possible and what there's a good signal to identify in the DIA data. So this is the ideal library, and as soon as we add anything to the library which is outside this orange circle and is in this kind of black or green zone here, this can only decrease sensitivity, right? So this is kind of the question that came up earlier. As soon as we start adding things that have no detectable signal in the DIA data, these are just channels where there's an opportunity to have kind of random matches and increase the false discoveries. And what you will actually find is if you do use these much larger libraries at fixed FDR, you're going to have to use a higher score threshold in order to achieve that same FDR. So this is to keep in mind when you're thinking about what kind of libraries to make. So we still do sometimes these larger scale libraries for kind of convenience, because it's nice to be able to just not have to generate a library again, but the sensitivity might go down. And also keeping in mind that we don't know, this circle is kind of an imaginary thing to some extent, and we don't know what that is. You might be able to figure out most of what's in that circle by doing kind of iterated analysis of the sample of interest, or looking at some transcript data, or having other evidence about what might be actually in the sample. But yeah, this is the take-home message here is the ideal library should be as big as what you can detect, and hopefully not bigger. Okay. So the summary about libraries is basically we need, or we don't need, but it's very helpful to have some prior knowledge in the form of spectral libraries for this peptide-centric analysis of DIA data. We have this protocol paper with everything written down in detail. FDR control at the library stage is very important because we don't want to propagate errors through the pipeline. Spectral library portability is possible, but with care. You need to evaluate whether your setup versus the setup, where the spectral library was generated are in some way comparable or not. And these retention times can also be, to some degree, you can transfer these around the place using this IRT concept. But again, the further away you get away from the chromatographic setup that was used to acquire the library, then the larger the errors will be in the prediction that you make based on this. Don't go for the biggest library available just because you can, because it's in many cases is just not going to help you, and this is because of this multiple testing burden that Olga was talking about. By way of kind of outlook, so now I've described to you the library generation protocol that we use by standard and have used for kind of 7 or 8 years in the lab. But there might be other ways to do this, right? So you might be able to analyze the data, and it basically looks like this, right? We generate some spectral library, prior knowledge, we do the peptide queries, and we generate peptide queries, and we query for these peptides in the DIA data. But there might be more to it than this. So Alexei Nazvishki later in the week will talk about this DIA umpire tool, where we're essentially generating the libraries in the DIA data. So I said you can't really search the DIA data using kind of standard database tools, database search tools, but if you try to deconvolute out these mixture spectra and make so-called pseudo spectra that represent the underlying peptides that were co-isolated together, then you can do a kind of a standard DDA database search, and DIA umpire is a tool that allows you to try and do this deconvolution. So this is going to come up from Alexei, and we also have a tutorial about this. And I think Mike McCoss will probably talk to some degree about directly analyzing the DIA data using in silico peptide generated queries, right? Just digesting the whole FASTA database and trying to see if they're there in the DIA data. And I'll let him talk about this. But one thing I think that we've seen is that methods that use the DIA data directly and aren't kind of library based definitely benefit from higher quality DIA data. And mostly what I'm talking about there is smaller precursor isolation windows. So as the precursor isolation windows go down, and the mixture spectra get less mixturey, less complex, then you can, I think, really effectively start to use these kind of library list types of approaches. Okay, great. So I'm going to leave it there. Are there any questions at this stage about libraries? Brandon, is this going to be about best replicate versus consensus? Yes. So I guess I would partially agree with that. So that totally makes sense. I think you will hear different opinions about that later in the week. So I guess Mike would agree with this. I think Alexei, for example, would not. And you could also distinguish between whether you would try to detect all detectable things in the library and then use some subset of that for quantification later. And Alexei's argument for this would be by taking things out of the library that are in the sample, you might make mistakes because now you have real signals in the sample that might map to something else, right? So you might have like something that looks like a peptide that looks like the thing that remains in your library, but the real entry, you've kicked it out already, and that would be a distraction. This is Alexei's argument.